Now I'm very proud to introduce you to our final speaker, Jean-François Laes. So he's our CTO, and I've known him since 13 years. We met when he was still working for DNA Vision, and then we were colleagues for a very short while because Jean-Paul kidnapped him really quickly as soon as he founded OncoDNA. So we lost him, but I got back to you, so time for you.
So good evening, everybody. So I'm going to talk about KDM because you heard this name now so a few times, and that does mean knowledge is driven medicine. So I will make a short introduction because you heard about sequencing, but just a short introduction. Where came all this next generation sequencing, all this data? So everything started in the Middle Age when I started my PhD in 1996, where we enjoy a very toxic gel and radioactivity, and we were very happy to sequence a gene within 6 months. Then a few years later, actually, there were a lot of invention that allows us to sequence faster the genes we are interested in. And actually, the throughput increase enormously, and then starting from just one gene, we were able to sequence more than a gene within 1 month, and then we were also able to sequence more genes. This leads actually to the first draft of the human genome that was achieved in 2001. It cost 300 million, sorry, I'm tired. And it took more than 10 years. And the final version was released in 2003. And then when it was done, actually, a lot of very clever people designed a lot of what we call new generation sequencing that allow us now to sequence a human genome within few hours for 1,000 euros. So you see how fast it was. But this is for research. And actually here, as you notice, we are talking about clinic, which is another story. So what was important for clinic is that the price is dropping because we are talking about patient, we are talking about medicine, we are talking about reimbursement, so the price has to be low. Also, people develop benchtop devices that all the biologists in the world could use within a simple lab, but that induce a very huge effect is the overload of data. Actually, the biologist was able to handle few genes, and now he has to handle a lot of data coming every day by those devices. And actually, what are the challenges that are facing the biologists nowadays? So first, you were heard that bioinformatics is a key, but actually there are different pipeline of bioinformatics which are not the same. You have special parameters, you have to handle this, and of course it will be too easy to use it with Windows. You have to work in black and white screen. Also, what about IT? I mean, do you have to invest in one computer? Which one? Do I have to use the cloud? Which one? And these are biologists. They have to think like IT people. Also, how to identify the biases? Because we know that every technology has biases. Every panel within those technologies have biases. Every lab using the same panel with the same technology have bias. So we have to identify those biases in order to avoid false positive and false negative results. Also, I mean, you might be an expert in DNA, but as you've seen, now biologists have to become also expert in RNA, in protein, microRNA, liquid biopsies, exosomes, how to handle all those different topics. Also, how to manage the information. Actually, because you receive a lot of data and you have to digest this information and to transform this information of something which is useful for the people. And also how to have something which is uniformized and centralized. As you've seen, you receive VCR from different labs, and you need a system that has to centralize this information in a comprehensive way. And also, this is the key, how to easily share this information between lab and between experts worldwide. So this is what we are doing every day since 4 years. Actually, based on our panel, actually we are able to generate very comprehensive reports coming from different machines and different also DNA from protein, from RNA. This is what we are doing every day. And as this has already been mentioned, data is not knowledge. Actually, you can have a lot of data, but what you are interested in is the knowledge coming from this data. So everybody is listening about big data, but this is not a big deal to handle big data. You just need more computer, but you need brain, and you need clever people and clever system to transform those big data into useful knowledge. The challenges for the clinician. Actually, very often they are receiving this information in different format from different pipeline, and actually they have to make a decision in very fast way. How can they then decipher this information, because they are not expert in all those fields. So they need that this information is already shown in a very comprehensive way. How to have access to this knowledge, because most of the oncologists are traveling worldwide, they are participating to meetings, so they have to have access at this information every time, in every place in the world. And how finally turn this data into useful for knowledge for the patient, because actually you can have a mutation. What does it mean in terms of biology? Okay, what does it mean in terms of treatment, and what does it mean in terms of patient survival and good quality of care? And that's why we have developed a huge database since 4 years, integrating 600 drugs. So these are a number, but actually we are integrated the variant, the clinical trial. We are also following clinicaltrial.gov, but we have to work on it because it's pretty complicated. Also, we have to integrate the different cancer type, the different information about the genes, and at the end of the day, to transform all this information in one and only comprehensive report for the oncologist. For all those reasons, we decided to create KDM, and more especially OncoKDM, because we are more specialized in oncology. What are the key features? I mean, for the lab manager, we are technology agnostic. As we already work with Salty, we can handle data from different sequencing machines. We can handle data from different other machines like NanoString, Sequenome, others. We are template agnostics. I mean, we can extract the information from data coming from DNA, RNA, proteins. We are also content agnostic, meaning that we have developed a huge database, meaning that we already have developed a useful information for most of the genes that are currently sequenced by the different platforms. And if there is something new, meaning that people are sequencing something which is not in our database, based on all the solution we have developed internally in OncoDNA, we can handle very fast this new information. You don't have any IT investment, you just upload the data in our cloud. I would not say don't have bio-IT people in your team, but I would say that use those bio-IT people for very complex problem and not for what is done in routine and can be handled by a computer. And also we are using a highly secure and highly ethic-driven environment, but I will come on this afterwards. So here, this is the workflow. So you send us the raw data. Actually, we perform a QC check because quality is really important. Because if you have bad quality, you will have bad results and therefore bad information. And we transform this into the VCF. And then from that VCF, we generate useful information for the oncologist and the clinician. And this workflow is true for any other technologies. We just start from raw data. As I mentioned, quality is really important, and often actually the machine generates a quality report which is not associated with the information from the VCF, that is not associated with the clinical information. So actually, by this system, we can gather everything within one report. And this is really important also for the biologist to know whether the sequencing was associated with good quality. As already mentioned, we have a huge capabilities to integrate information coming from different devices and different templates. This is something we routinely do since 4 years. We also automatize a lot of things, but I mean, we still have to be flexible and taking into account that actually the biologist will transfer the data to his collaborator or her collaborator or the oncologist. So actually, we have to let the biologist having the flexibility to change or to adapt the text we wrote. So we facilitate their life, but I mean, at the end of the day, they are responsible for the information. So we nevertheless let the possibility to the biologist to change our text. If they not agree or they want to write something else, they can also hide some variant because from ethical point of view or to patented point of view, they can hide some variant. But they have to do that in an objective way, meaning that we also provide them with what we call a viewer. So when you have the raw data, actually you can use a viewer to check the quality of your raw data to be sure that the mutation you have identified is a true positive. And we offer this possibility too. So we automatize everything, but at the end of the day, you are still the proprietary of your own data. And what is important is also the sharing of the report. So we send you the report, and then you validate, and then the report is shared with the different collaborators you implement into the platform. And as soon as you did that, actually all the raw data are removed from our server, meaning that we don't keep any link between the patient and the data. What we keep is the information, meaning that, for instance, you have patient X that has a breast cancer. This mutation has been treated by this treatment. After the validation, actually, we will not have the link with Mrs. X. What we will have is really the information, meaning that in our database, we have a case that contains that mutation, treated with this treatment, and receive and had that outcomes. And you are not working alone, meaning that, of course, every patient is different, and especially the evolution of every patient is different, meaning that actually, when you have a patient, you have something that is rare. And you have to gather all this information worldwide in order to have what we discuss about e-learning, so the useful information shared with different patients that will improve the outcome of the patient that you analyzed. So it's really important to generate a lot of data and a lot of knowledge that can be shared through our platform in very anonymized way. So for the oncologist, I mean, we can provide him with some information about the quality of the pathology. This is a summary that we routinely provide the oncologist with. I mean, sometimes they don't have the time to read everything, and they have to make a decision within 20 minutes. So we have to provide them with a very comprehensive and accurate summary. You also have the information about clinical trial. And here, I mean, like mentioned in the previous talk, more and more patients are monitored through liquid biopsy. And actually, we have also to include this information within the same report to see the evolution of the disease and the evolution of the different mutations that are fetched within those liquid biopsies. Same for the oncologist. Already mentioned, this is really important because they are making a molecular board. And actually, they are gathering in order to discuss about cases. But we have to provide them with a very comprehensive way to show all the data and web-based, because a lot of them might be abroad. So based on this platform, they could be able to discuss about the case. Of course, what is discussed is secret. It's a medical secret, but what can be implemented is the conclusion, meaning that after the discussion, we decided to treat the patient with this treatment. And then this will goes to the report, meaning that the next time they will again gather together in order to discuss about this case, actually they will have the last information about the treatment. Like it is the case for the moment, you have a verticality of the follow-up of the patients. So all this is actually has been built to achieve one and only priority, is the treatment option for the patient. So I will call about the OncoDNA project. So OncoDNA, so very strange name. So at the beginning, people don't like it because I didn't explain why I choose this name. So I will explain to you. So it's coming from the idea of OncoDNA project, which has been imaginated by a Belgian guy called Paul Otley in the 19th century, and actually is the first idea of internet. Of course, there were no computer at that time, and actually he realized the paging, you know, the famous Larry Page from Google, actually it was already invented by a Belgian guy, but in small paper. So you have a big room with small paper saying, oh, this is interesting, go to room number X, page number Y, and then you have actually an internet in paper. So actually this guy invented, and it's a Belgian guy that invented internet, or actually the concept of internet. And that's why I call OncoKDM, and when I explain that, people like it very much, especially the people from Belgium. So what is the project? So actually we were granted, the idea is to speed up the deployment of our KDM and to be at the forefront of the science. Because as I mentioned, actually to have a lot of data and a lot of knowledge is the key in order to really provide intelligence to the clinician. This has been financed by the Walloon region at 55%. Jean-Paul said 9 million, I said 8, so it's 8.5. So the first phase will consist in identify a key center in Europe that are performing NGS, but not only, and to work with them in a collaboration way in order to provide them with our solution and also to learn from them about the issues, the way they want to show the data to the clinician. After we'll have a phase 2, which will be after the evaluation, it's the real work. So first we'll have to work on sample where we already know the results. This is really important because at the end of the day, the biologist and the oncologist have to trust us because they will base some decision according to our results. So this is really important. We are very keen about quality of the data and the report. Phase 3 will be, okay, we have made all the samples, everything were fine. So how to make publication, how to go perhaps further in this collaboration. And the phase 4 actually is something which join what we call the e-learning machine. So actually we would like, so don't go to US, and we will please to hire some people in OncoDNA. So we already work on this topic, which is called the patient matching. So based on the knowledge from the data, actually we could identify the most similar patient in our database. So imagine that you have a cancer patient, has a breast cancer, has this mutation, has been treated by this treatment. Actually, we could be able to find in our database the most similar patient, meaning that you will have the information that this patient also had this mutation, has been treated by this one, and what were the outcomes. But to generate intelligence, you need knowledge, and to generate knowledge, you need data. So we have to have the whole pipeline. So in conclusion, so KDM is not integrating the information from RNA, but also RNA and protein. I mean, it's like if you want to explain the world with only one element. It works 2,000 years ago, but actually we need the whole element in order to really understand the world. It's the same with tumor. You need to know all the elements to really understand the tumor and its evolution. it's unique customizable platform, but actually with strong commitment of quality. But you know that every platform has some bias. You have to fit a bit the parameters for the algorithm. So actually, yes, we work on a very strong core pipeline, but we have to adapt and customize a platform and our algorithm to fulfill the expectation of every platform. And also very important is that it's a web-based solution without any IT investment and available worldwide, meaning that actually the machine are generating sequence, you don't have any investment, and actually it's working for you, and you can look at the data anywhere in the world and on holiday if you are very brave. And it's a unique platform where you can share the results. So I assist to different molecular boards, and it was slides in different formats. There are some people who were not there, not able to attend. And actually with our solution, actually it's standardized. You can click on everything, and you can also discuss with anybody worldwide. So even if your expert is at the ASCO meeting, actually it could nevertheless participate to your molecular board. And here, because we are ready to launch this, it's OncoKDM Discovery. So what is OncoKDM Discovery? Actually, it's a platform that has been built in order to let anybody worldwide to test our solution. So here are the screens, but this has been already shown by Dr. Sonia. Yes, sorry. So you see here we are showing quality, next generation sequencing, treatment. Also, I mean, we also provide you with a different interpretation of what we found, and everything is always based on what is available on the web, meaning that you can check every information based where our texts are based. So you don't have to trust us, you can check everything. Everything is open. Also, we go really into detail. We are not just simply analyze gene by gene. We analyze down to the exome level, down to the base level, and quality of the base. So we are really going deeper and deeper and deeper in the quality of the data. So KDM, which will start today officially, as I mentioned, is available worldwide for anybody. Of course, you need FASTA-Q file, and it's for 5 samples, small panel, and for some only few cancer type. And I would like here to thank people that work very hard to make this available today. So let's start. It's online, and I thank you, and I hope you enjoy the drinks for friendship.
Questions for Jean-François?
You were giving the job of oncologic geneticians to oncologists. What about giving the jobs of oncologists to general practitioners? This means giving also access to general practitioners to your database, because this is what in the end will happen, that the knowledge will trickle down and that the general practitioner will order, let's say, the liquid biopsies and the follow-up of the DNA. And I have a practical example of this. I'm helping a USA patient, and I'm trying to convince her to use OncoDNA. But now she said to me, yes, but I will first try a kind of cheap system. It's called 123. Well, it's a special, very simple name, but it's a genetic testing that is offered in USA for $200.
Yes, it's 23andMe.
Yeah, but
yes.
And so my question is, could someone with this kind of external genetic information also enter this into your KDM system?
Yes, yes. Actually, Actually, we have a lot of debate internally. So for me, KDM is for any problematic. For the moment, we are concentrated in oncology because this is where we are expert in. But I mean, in the future, we could collaborate with other experts in different disease, also to provide them with this platform. But the expertise will be theirs. But I mean, this is the future, of course. We have to focus of what we know. But there are no limitations as far as I see.
And you will also make the platform available for general practitioners, or is it for the moment only?
Yes. Just we are only 50 people, but yes, in the future, when we will be 200, that will be with pleasure.
A comment maybe from the oncologist in the room?
From me, just congratulations on the initiative. I would be very keen to hear about real everyday life feedback, so to say.
If you can test it, I will be pleased.
Yes, and I would like, in response to your question, I think general practitioners can have this information, but the patients are being treated by medical oncologists. And as you said, each patient is unique, and the evolution of each patient and side effects. And always you have this information, but then the manner you apply to your patient, it's unique, and it depends on how it's being treated, which treatments they have received. So I think that medical oncologists are important and it's needed. And I think, in my opinion, for me, it's no problem that other clinicians could see all this information, even though maybe they cannot have all the information and interpret all those results. In my opinion, and congrats as well with this. It's amazing. I think it's where we have to go, is to share all this information, and in order, as you said very, very well, it's not only data, it's not only knowledge, but we have to do this in order to more patients could benefit. Perfect.
Thank you very much. Muchas gracias.
De nada.